to another video. Today we want to talk about the Golgi apparatus, and I guess you're all familiar with it, but today we want to talk about some details. So the Golgi is a cell organelle within eukaryotic cells, and it's responsible for modifying, packaging and transporting proteins. So it's called Golgi apparatus, or also Golgi body or complex, and you might also hear just the term Golgi. It all means the same. Usually it consists of about 6 cisternae, which are actually flat porches that are flattened and stacks. In some organisms, however, the number of cisternae might be as high as 60. The Golgi gets supported by microtubules and is stabilized by matrix proteins. It can be divided into 3 compartments. So we have the cis, the medial and the trans Golgi. And here you see the cis and here the trans side. They are responsible for sorting lipids and proteins. And lipids and proteins can be received from the cis side and are released at the trans side. Actually, there's a specific connection between the endoplasmic reticulum and the Golgi, which is the so-called vesicular tubular cluster. And it's unclear how protein transport from cis to trans Golgi works in detail. So the Golgi is, compared to other organelles, relatively large. So it was discovered rather early, which is at the end of the 20th century, by an Italian physician, which was called Golgi, and thereby the name. So even though at first there were doubts about the existence, it got confirmed at the beginning of the new century, as better microscopy techniques were available. So the subcellular location, so where the Golgi is within a cell, can actually vary between different eukaryotes. So in mammals, like ourselves, it's usually close to the nucleus, while for example in yeast, it's distributed throughout the cytoplasm. And in plants, the stacks are not concentrated. And here the organization depends on actin rather than microtubules. So all have in common, however, that the ER and the Golgi lie close to each other. And now you've heard the term ER actually for a second time, so if you're not completely familiar with this organelle, or if you want to hear some details about it, there's a video about it which is linked above. Now, the essential functions of the Golgi involve the processing of lipids and also proteins. It can act, for example, carbohydrates to modify molecules. It receives from external sources like the ER. And thereby it can create molecules like cholesterol, which is the basic component of the cell membrane and therefore highly relevant. The Golgi can also add labels, which are responsible for bringing the right cargo to its destination. So for example, mannose 6-phosphate. So, which you see here. So thereby the Golgi is an essential part of exocytosis, where proteins get secreted. Now, let's take a look at the 3 types of proteins that get secreted. So we have first cell surface proteins like cholesterol that was just mentioned. Here you see the molecular structure. Then we have lysosomal proteins, and here especially the enzymes have to be packed into vesicles because those could otherwise harm the cells. And then we have secretory proteins, and this can be, for example, hormones. So if we want to excrete those secretory proteins as exocytosis, this can be either regulated or unregulated, and as a stimulus for excretion, a hormone would be a possible stimulus. Now, and if we take a look here how the fusion of a vesicle works, so here we have a vesicle with a cargo within, then it gets closer to the cell membrane and starts to fuse with it, and ultimately it's completely fused with the cell membrane and releases the cargo to the exterior. As already indicated before, the ER and the Golgi are not only in close proximity, but they also work together closely. So we have vesicles from the ER that go to the cis side of the Golgi, and then they get modified. Actually, both are involved in vesicle transport, which is of course essential as they produce and transport substances that are required throughout the cell. And according to the respective signal sequences, which are usually a sequence of amino acids, they can be sent to the desired location. This was a brief overview of this highly relevant organelle. If you like this video, don't forget to give it a thumbs up and to subscribe to the channel to see weekly uploads on scientific topics. See you in the next video.